Chapter 8, Cells and Their Environment. So before we start this chapter, let's do a little bit of a review about what's going on from last chapter. So in your groups right now, I'd like for you to name at least 5 different organelles and their functions. I would also like for you to list the 4 main types of biomolecules. We learned about those in chapter 2. You can pause the video while you go through those 2 questions right now in your groups. Okay, so section 1 is going to be talking about the cell membrane. So the cell membrane is an important organelle that is found in 1. Prokaryotes and eukaryotes. Remember, prokaryotes are single-celled organisms, eukaryotes are multicellular organisms and more complex organisms. This is a flexible membrane, also known as the plasma membrane, that surrounds us. It's found in both plants and animal cells. However, the cell wall is the first layer of protection for plants, and then the cell membrane is underneath of that. So the cell membrane is really important for regulating homeostasis. Homeostasis is maintaining a stable internal environment. So why would we need to regulate or maintain homeostasis? I'd like for you to pause the video right now and in your groups think of 3 reasons why you would need to maintain homeostasis within a cell. Okay. So the cell is able to regulate the materials that move into and out of the cell. This helps to maintain that homeostasis. 1, we want to get rid of waste products or toxins that are produced by the cell. 2, we want to make sure the cell is taking in the nutrients it needs so it can survive. And we also have some products that are made by the cell that are needed in other places, especially in eukaryotic organisms. So those need to be transferred out of the cell. Okay. So here's why we need to maintain homeostasis. 1 is structural support of the cytoplasm. Cytoplasm is made mainly of water, and if you don't have enough water within your cell will actually shrivel up. In plant cells, they have a large central vacuole to maintain this to help to maintain turgor pressure, which also maintains homeostasis. But our cytoplasm is really important for that. If you become too dehydrated, you can your cell will shrivel up and die, but you can also suffer from things like, as a human being, such as hallucinations and basically dementia, which is also a stage really close to actual death of dehydration. We also use the cell to recognize foreign materials such as bacteria. We want to keep those out, or waste products or toxins within the cell. And cells can communicate with one another. Remember, in a eukaryotic organism, it is important for cells to be able to recognize each other and to be able to communicate between different cells. Now, this cell membrane is made up of what we call phospholipids. This is a double membrane or a double bilayer, but the phospholipid itself is really important because it has a polar head and a non-polar tail. So that's really kind of neat because the polar head will attract water, but the non-polar is a fatty acid, and it, like any lipid, it repels water. So it doesn't actually allow water to transport into and out of the cell freely. The only thing that's able to go through the cell membrane without a protein assistance is really tiny non-polar molecules. And so it's cool because we can attract those water molecules, so we get them close, or we pull them into the cell or towards the cell, but we can't actually put them into the cell. This helps to maintain what we know as an isotonic state, which we'll learn about in the next section. But this allows for our cell to make sure that it's not going to explode at any moment. So those are the 2 regions. Also, when we look at the phospholipid, we have a phosphate molecule, but the 3 most important elements that we find in here are carbon, hydrogen, and oxygen, which are found in all of our biomolecules. Okay, and so we can see that here. Now, as we just talked about, we have a hydrophilic and hydrophobic. So we don't only call them polar and non-polar, but we call them hydrophilic and hydrophobic. Water loving, again, that head is bringing in the water molecules towards the inside of the cell and towards the outside of the cell. And then the non-polar hydrophobic or water-fearing portion, which is actually your fatty acid, is not allowing those to go into or out of the cell without the regulation of a protein. You'll find out that not only these phospholipids or fatty acids are making up our phospholipid bilayer, but we have these proteins that are really important to maintaining structure, sending signals, and allowing for certain materials to pass into and out of the cell. Again, always to maintain homeostasis. So here we can see the structure of our lipid bilayer. It's bi means two. And so we have two layers occurring here, hydrophobic inside and a hydrophilic outside. And so we have our water-loving outside and inside, and our water-fearing on in the middle. And this is for the sheer purpose of making sure that we only have the materials going into and out of the cell that are actually needed to be there. It prevents a barrier, and so we can only, this structural barrier will only allow, again, small non-polar molecules to go into and out of the cell freely through a process we call diffusion, which doesn't require any energy for this. Okay. And so the barrier, the phospholipid barrier, we can see some ions. So what is an ion? What constitutes an ion? So I want you guys to think of at least 3 ions that our body would need to go into and out of cell, or to talk about what would be an ion. You can pause the video right now and have that discussion of what is an ion. So an ion would be like sodium. It has a positive charge or a negative charge to it, and that ion allows for our bodies to send electrical impulses from one cell to the next. It will also allow for us to send certain signals, or to be able to do things like a muscle contraction, relaxation, such as the sodium and potassium ions. Allow our muscles to contract and allow for them to relax. So this barrier is what we call semi-permeable. Why would it be important to have a semi-permeable membrane instead of a completely permeable? So what does that mean to be semi-permeable? Semi-permeable means allows some things to go into and out of the cell, but it doesn't allow for everything to go into. So why don't you pause the video right now and talk about why you would want a semi-permeable cell membrane. Okay, as we talked about before, a membrane is really important to maintain homeostasis, and so we have some proteins which are needed for this. So we have proteins that face inside, proteins that face outside, and we have these proteins that will go all the way through. But the most important thing is we have what we call a peripheral protein and an integral protein. An integral protein is a protein which is permanent. It's on the cell membrane all the time, no matter what. And then we have a peripheral protein. These are only there temporarily or partial, and they're usually proteins that will face on the outside of the cell. And so we have 4 main types of proteins which attach to our cell membrane or plasma membrane. One is a cell surface marker, one is a receptor protein, an enzyme, and a transport protein. Okay, a cell surface marker is the first one we're going to talk about. The cell surface marker is usually a peripheral protein, which means it faces outside of the cell and can be detached. Its total job is to identify what the cell is. It's like a name tag for the cell, telling us what it does, what it is. It usually will have not only a protein but have sugars attached to it, which are carbohydrates. These are called glycoproteins, and they work with the carbohydrate works with the protein to kind of act as a name taker, identifying marker for that cell. We'll have a receptor protein. The receptor proteins will recognize and bind to substances outside of the cell. They usually will go all the way through the cell membrane, and so they are able to sense what's going on inside of the cell and what's going on outside of the cell. And so if there's changes that are occurring outside of the cell, it will make the cell aware so it can prepare the inside to make those same changes, such as if we have a high salt concentration outside the cell, it'll actually move water from the inside of the cell out of the cell to try to maintain homeostasis for the entire body. And so that's what those receptor proteins are. They're changing the internal environment of the cell based on what's happening outside the cell. Okay, we have enzymes, which we are already familiar with. We've already talked about enzymes. Enzymes are catalysts. Catalysts, these are temporary proteins, and so we call them peripheral proteins. They are found on the outside and the inside of the cell. They're typically inside, and they will assist in chemical reactions. And so their job is to lower the activation energy needed for a cell to start a chemical reaction. So it basically allows for the same chemical reaction to occur, but at a faster rate, which means it takes less energy. And you guys should know what the body's energy source is. That is ATP, which is a nucleic acid, not a protein. And the last type of protein that we're going to talk about is a transport protein. Transport proteins are actually really important because they transport materials into and out of the cell. Now, transport proteins can be both an integral protein, which means it's permanently attached to the membrane, or it can be a peripheral protein. Depends on what type of material it's transporting. And so one type of transport protein would be a channel protein. This channel protein allows for materials to move into and out of the cell. It is an integral protein, it's always attached there, and that would be an example of a water channel. Water, again, as we said, is a polar molecule, so it can't travel freely through the lipid bilayer. And so you have to use a channel protein to move water into and out of the cell. And the reason we want to do that is because if we get too much water inside of our cell will actually burst or explode and it will kill the cell. But if we let too much water go out of the cell will shrivel up and die. So we want to maintain that homeostasis through that transport protein. We also have some active transport proteins, which are called pumps. Sodium potassium pump would be an example of that, which are integral proteins, which are there all the time. And they allow for the movement of sodium and potassium ions into and out of the cell, which allows for muscle contraction and relaxation. If we don't have that, then your muscles will contract and they can't relax, aka you get a huge cramp in your leg. The other type of protein that's kind of cool with this transport protein are the proteins known as vesicles, and we use those for endocytosis and exocytosis. Those are just peripheral. They're there temporarily. They actually look like a little Pac-Man for endocytosis, where they open up, they'll climb around something, and they'll bring it into the cell. And then we have exocytosis, where we have this little vesicle attached to it, and it kind of like vomits whatever back out of the cell. And so those are the four main types of proteins we have. So you guys are going to have a homework assignment on Classroom. That is already up, so that is due tomorrow, and you are going to be making what we call a fluid mosaic model of the cell membrane. The fluid mosaic model is a model that shows a three-dimensional structure of our plasma membrane or cell membrane using our cell surface markers, receptor proteins, enzymes, transport proteins. All those are going to be in there, and you're going to have some questions you're going to need to answer on those. So tomorrow you are going to be doing a group activity, basically going through and talking about all these. Alright, talk to you later.